GenScript ProBio GMP Manufacturing Center contains GMP Warehouse, bulk production area, and high-standard QC laboratory, which meets the FDA and NMPA regulatory requirements. The first floor, GMP Warehouse. It is divided into room temperature storage, raw material cold storage, intermediate product cold storage, finished product cold storage, refrigerator area, rejected product area, and sampling room. Optimized circular layout, logical traffic flow. The warehouse implements 5S, refined management, and business operations are orderly and efficient. Sufficient and complete storage space. Room temperature storage of 800 square meters. There is a 100 square meters cold room, temperature 2 to 8 degrees, which is divided into 3 independent units for raw material, intermediate product, and a finished product respectively. Refrigerators in different temperature zones, minus 20 degrees, minus 40 degrees, and minus 70 degrees. Warehouse is managed by partition. Materials are stored in the corresponding areas, for example, quarantine area, qualified area, and rejected area according to different quality status. The areas for different status are clearly marked to prevent confusion. The second floor, Antibody GMP Production Factory. The design and construction of the Antibody GMP Production Factory on the second floor meets the requirements of China, the United States, and Europe, and the GMP management meets the NMPA and FDA clinical Phase 1 and Phase 2 regulations for biological drug projects. The factory is equipped with 2 single-use bioreactors of 500 liters, 6 single-use bioreactors of 200 liters, and 2 downstream purification lines. The whole factory enables unidirectional flow. The design annual production capacity is 80 batches. It can provide customers with timely and reliable GMP production services, which can also meet their professional and customized production needs at the same time. The periphery includes sterilization room, cleaning room, and laundry room, which also function under the unidirectional flow design, reducing contamination risk. Thermo Fisher Cyto Extra gas phase liquid nitrogen tank in the cell bank stores biological samples with high density to avoid cross-contamination, which can send SMS and email alarms to ensure safe storage. Upstream seed culture room is equipped with Enforce carbon dioxide shaker and ISCO BSC. Upstream cell culture room. Thermo Fisher 200 and 500 liters single-use bioreactors. They are used for monitoring cell culture process, online monitoring parameters, for example, temperature, pH, dissolved oxygen, and ventilation, etc. They can be used for technological process development and large-scale production of batch culture, fat batch culture, and a perfusion culture. Fully single-use system with capacity of 2,200 liters, in compliance with NMPA and FDA cGMP requirements. Cell culture room is equipped with wave bioreactors for small-scale seed train. Upstream lines have IPC rooms to test the parameters during cell culture and harvest rooms. All the equipment is from global leading suppliers. The purification room is the key room for the entire downstream production. The process in the room includes affinity chromatography, low pH, neutralization, intermediate depth filtration, anion and cation chromatography, etc. And finally, the nanofiltration process steps. The chromatography system consists mainly of the ACTA-ready single-use system. Nanofiltration mainly uses VF3 single-use system and manual operation mode system. The single-use system can mitigate the risk of cross-contamination while reducing the downtime between batches and improving production efficiency. Together, they meet all the demands of purification and virus removal processes. Ultrafiltration and bulk filling room. Single-use ultrafiltration system can realize online monitoring. The ultrafiltration and bulk fill room has an independent air conditioning system. The ultrafiltration equipment consists of millipores TF3S disposable ultrafiltration system and a manual operating system. The process of adjuvant adding and bulk filling are carried out after ultrafiltration. Downstream buffer solution is prepared with low contamination risk, which is featured with sartorius mixing systems, backholders, Italy Fatigari autoclaves, Steris 480 PG washers, laundry washing machines and dryers. The bulk production plant adopts the unidirectional flow design and the fully closed system and process. Each production line is designed to be physically separated. With an independent air conditioning system, the design allows multiple projects to be operated at the same time without cross-contamination risk. The third floor, GMP QC Laboratory. The physical and chemical laboratory is mainly for testing basic physical and chemical indexes, including proton concentration, pH, turbidity and osmotic pressure, etc. ELISA and qPCR laboratories are used for impurity detection during product process. The instruments are all inspected and calibrated. All experimental data are strictly managed in accordance with regulations of each external regulations. In addition, the qPCR pretreatment room adopts a full air exhaust and supply system, which can effectively prevent cross-contamination during sample processing. QC is currently equipped with stability sample room to study product stability. The series of binder stability test chambers, which have completed IOPQ and calibration, complying with the requirements of ICH and worldwide pharmacopoeias. Meanwhile, each chamber is equipped with an EMS system to ensure real-time monitoring of temperature and humidity. The balance room is equipped with a number of sartorius high-end, high-precision electronic balances that have been strictly qualified, which can meet the requirements of various weighing tasks. The chromatographic room is used for biological HPLC and CE analysis. The laboratory is equipped with world-class analyzers, for example, HPLC, UPLC, CE, etc. At present, all instruments have been calibrated, qualified, and completed the computerized system validation. The computerized system is strictly managed according to worldwide regulations to ensure the authenticity, integrity, and traceability of the data. At present, the QC laboratory has been equipped with appropriate facilities, equipment, and instruments, as well as trained and qualified personnel, which can effectively and reliably complete all quality control-related activities. High construction standards, leading capacity in the industry, perfect technology transfer process, zero crossover production lines, fully single-use manufacturing system, international quality system. GenScript ProBio GMP Manufacturing Center will develop together with you.